Dr. MP, madam?
I hope I'm audible.
Yeah, you are audible. You are,
Yeah,
you are visible, audible. Now
great.
put
Thank you
the
so
slides,
much.
madam.
Yeah, sure, sir. I'll just
PPT.
start presenting. Yes, sir. Hello, everyone. First of all, I would like to uh, congratulate the panel and organizer for organizing such a beautiful workshop and symposium. And I want to thank also everyone for giving me an invitation over here. Today I'm going to talk about exploring the impact of SARS-CoV-2 virus infection on amyloidosis development. I'm Dr. Rimpi Kaur Chauhan, Assistant Professor at Department of Biomedical Sciences, Acharya Narendra Dev College, University of Delhi. We and our team in our college are kind of exploring how cardiac amyloidosis basically is affected. Now, first let's get and understand about what is amyloidosis. We all know it's a disease which is characterized by accumulation of misfolded or aberrant proteins in the form of amyloids, which hampers the functioning of the normal cellular physiology as well as causes toxicity within the cell. Over the years, we have realized with the help of various experiments done in humans and mice that it is an age-related phenomena as well. To that extent that we consider it as a universal disease of aging. So as the age increases, amyloid accumulation has been seen to increase with multiple folds. What causes this amyloidosis is considered to be or attributed to be majorly the mutations within the proteins, which ultimately leads to production of misfolded proteins, which keeps on getting accumulated over the course of time and enhances the generation of what we currently know as amyloidosis phenomena. These kinds of diseases occur in your brain tissues, cardiac tissue, as well as various other parts of the body, which you cannot even consider. Like, for example, insulin aggregation leading to diabetes is also commonly known now because of the amyloidosis phenomena. So, which brings us back to the recent past, we have seen emergence of viral disorders. That viral disorder was SARS-CoV-2, COVID-19. The whole world was impacted by it. And when I say the whole world, it is also an age-dependent phenomena, and we saw a majority of elderly or senior citizens were also majorly affected by this disorder. And over the course of time, the various studies came up which have shown that SARS-CoV-2 infections, in fact, is kind of leading towards there in those cases, or maybe in the cases of long COVID, or in the patients which have been infected, has shown no particular genetic history of amyloidosis formation, was also showcasing presence of amyloids or presence of protein aggregates within their body, which led us to the belief that maybe this particular viral infection has a role to play in amyloidosis. For the sake of understanding, we restricted our preliminary studies on cardiac amyloidosis. We first wanted to understand maybe what's happening over there. Now, first of all, let's understand the mechanism of SARS-CoV-2, how it enters our cell or our body. It utilizes the major surface proteins involving spike protein, envelope proteins. It interacts with majorly ACE receptors present on the surface of our cells, enters inside our body, releases its genome, and the genome is then utilized to create its own protein and carry out the lytic cycle that we know about. Now, there were other studies which were coming in as well. These kinds of studies were showcasing, in vitro studies which were showcasing, or some scenarios, bioinformatics studies which were showcasing that these spike and envelope proteins was found to, interact with serum amyloid A, another serum protein, and was leading to formation of, or had the capacity to form amyloid fibrils from this particular protein. At the same time, downregulation of ACE2 receptor and upregulation of spike proteins within the lungs were after COVID infection was found to lead to formation of amyloidotic microclots within the vessels. On the other hand, there was a large pool of cases coming up that in the long COVID, there were accelerated rates of amyloid formation. And then we also saw that after vaccination, certain individuals were showing large amount of clot formations and protein aggregation, which led us to an hypothesis. We believed that because we know that amyloidosis generally happens or starts because of the structure being affected. It's the gene mutation leading to the misfolded protein leading to amyloid formations. So maybe, and now we know so far that certain amount of proteins, of surface protein specifically of virus, has the capacity to interact with human proteins. So maybe there is a direct cause and effect relationship of this interaction, which is leading to certain proteins becoming more amyloidogenic in nature. So let's understand this biochemical mechanism, because we do have a large amount of, already within our body, we have a large pool of proteins which are aggregation prone, or we refer it to as amyloidogenic in nature. So we started our study. We first took up spike protein, envelope protein, membrane, and nucleocapsid protein. These four proteins are present on the surface of the virus. Then we scrutinized the viral proteins. Then after that, we scrutinized our human cardiac proteins, which are already known to participate or have been seen to form aggregates or amyloids or amorphous aggregates in the case of cardiac amyloidosis in patients and leading to fatality. So in general, we knew any kind of mutation or any kind of trigger within these proteins is bound to trigger a large amount of reaction. So ideally, in a normal person, where their genes coding for this protein is perfectly wild type, amyloidosis never occur. Only in the patients where the gene coding for these human proteins is mutated, or because of XYZ features which leads to aggregation or changes the structure or leads to misfolded in the protein, can only trigger amyloidosis among these proteins, otherwise not. So we wanted to understand, maybe there's a chance that if the viral proteins are interacting with these human cardiac proteins, then are they also increasing the aggregation potency of these particular human cardiac proteins? So to understand all of this, we carried out a bioinformatic analysis. We explored all the potential interactions between SARS-CoV-2 structural proteins and human amyloidogenic cardiac proteins, and assessed the potential role of these interactions influencing the amyloidogenicity of the cardiac proteins. So the first thing we did was we carried out a preliminary docking studies. We tried to understand what all possible interactions could be there. So we had 9 human cardiac proteins, which we had narrowed down to. Tried to interact. We see other interaction with the 4 surface proteins of the virus, and we found, to our surprise, that all amyloidogenic cardiac proteins were showing good amount of binding energy or binding interaction towards the viral structure proteins. And in fact, if you look at the B category, that was the binding interface, even the interface area was pretty large to not consider them as something which is to be taken seriously. The other interactions, hydrogen bonding, ionic interactions, and van der Waals interactions were seemingly high as well in majority of these. Since in comparison to spike, membrane, and nucleic acid, envelope protein was the one which was showing strong interaction with all of them, while in the case of spike, we can see it was not strongly interacting with gel. So, say there were certain cases where the interaction was not found. Envelope, on the other hand, the envelope viral protein was showing strong interaction with all the cardiac amyloidogenic proteins. So we then, just to understand or have a better understanding of this particular interaction, and also investigate the stability of the dot complexes, we streamlined our study to investigate the interaction between the monomeric form of envelope protein and human cardiac proteins. Why particularly monomeric? So envelope protein can exist in both multimer and monomer formation, and all the forms have been shown in the past to perform or being equally important and present in equal amounts on the surface of the viral protein. And utilizing a monomer protein allows us to have a better understanding of what is happening in terms of interaction within our protein, considering it's a in silico study. So we again carried out our interaction studies with the monomeric envelope protein. As seen earlier with the multimeric as well, the monomer was also found to show good amount of interaction, very large negative binding energies, solvation and electrostatic and van der Waals interactions were also pretty high. So basically showing that all the myelogenic cardiac proteins were spontaneously willing to bind with monomeric form of the envelope protein. We then carried out the simulation of these complexes. After doing that, we found the simulation, the RMST profile was stabilized after the 10 nanoseconds for almost all of them, showcasing that the stimulation performed was done in an efficient manner. After the simulation, the pre-simulated and post-simulated interaction, the interface area, in fact, was found to be enhanced, showcasing that the docked structures which have been earlier found, they were highly stable, and that's why even after simulations, they are still stable. They are still showcasing interaction among each other. Then our next goal was to understand how this interaction is affecting the structure of the human cardiac proteins. And we saw that the solvent accessible surface area was found to be, almost like, for example, in the case of apolipoprotein A1, it was found to be almost normal. ANP and fibrinogen was found to decrease. So we were looking at, so what happens is, we have your hydrophobic amino acids within the core of the protein, and hydrophilic amino acids are generally on the solvent area, near the solvent on the outside. This kind of conformation, majority of the time in case of monomeric proteins, allows the protein to remain stable and not aggregate. Now, when the proteins start opening up, their solvent accessible surface area starts increasing. That implies that the protein is opening, or the structure is changing or altering in a very different manner, where it is now has more access to solvent. When that happens, it also inclines us to believe that maybe the change in the structure is to that can lead to opening up of the structure in a format that the hydrophobic amino acids are getting exposed to solvent. And that is never a good thing in cases of if any possible interaction is not in the vicinity in the phenomena. Because the moment your hydrophobic amino acids are exposed to the polar solvent, they don't want to undergo that interaction. To bypass or to get safe from that, they will prefer, in fact, to interact with another protein with, and form a hydrophobic interactions with them, which leads to a ordered formation of one protein interacting with another. Earlier, they were not supposed to. Now they're doing it, leading to multiple proteins doing the same phenomena of interacting with each other.
Please complete.
Okay, leads to the formation. Yes, sir. 2 minutes more. Leads to the formation. Yes, sir. I hope I'm audible. Just 2 minutes more, sir.
Thank you.
Okay. So leads to the formation of protein aggregates. So in the case of this particular case, we found atrial natriuretic peptide and fibrinogen alpha to in fact increase in the SASA. So ANP and fibrinogen alpha was recognized to have that structure altered to a very larger case. Similarly, we also analyzed the hydrogen bonding profile and found that apart from gelsilin and transthyretin, almost all of them were showing strong hydrogen binding interactions. And ultimately, we checked whether, because of the changes in these structures, is there an enhancement in aggregation propensity? And we found apolipoprotein A1, gelsilin, ILH, transthyretin, and beta microglobulin. All 5 of them were showing huge change in the structure, which were leading to the aggregation propensity being enhanced. So these are all wild type proteins, which after the interaction with surface proteins of the virus were showcasing huge change in their structures, and also quite a high escalation in their aggregation propensity as well, which leads us to have a confidence that maybe if we look into these studies in vitro and in vivo, it will give us a better idea about the mechanisms of how protein amyloidosis takes place in general, and how we can in fact model these kinds of, with the help of viral infection, we can model these kinds of studies within the mice and others and study amyloidosis without wasting a lot of time in creating an age-related model. I thank all of you and the principal and my collaborators, Dr. Rachna Pandey, DPT Star College, and our mentors. Thank you very much for giving out your time. If you have any questions, let me know.
Thank you. Thank you for the nice presentation on the bioinformatic tools, particularly using the SARS envelope proteins on amyloidogenesis. Very nice talk, madam. Thank you.